Good. So everyone can see this other computer here. So before I start, this is a Linux computer. And in this Linux computer, we have, we go down here, we have a GeForce NVIDIA GPU graphics card. So PSORT also supports GPU, and especially it will be really helpful to have a GPU. So if you have a Linux computer and a GPU, life will be much, much, much easier for you in using PSORT because everything will be much faster. So if you have such a thing in your lab, I will highly recommend working in this setup. So let me go ahead and run PSORT. So since I already installed PSORT on this computer, by just typing PSORT, I should be able to run the software. Anybody have problem running the PSORT, or all of you can basically see this window? Is that correct? Cool. Good. And the first example that I'm going to cover here is a very simple recording. So sample data. In our lab, we are, when you load a data set, the first thing that will ask you is that if you want to load a common average data or not. So in our lab, our main signal and our common average are two separate signals. That's why we can load the common average. If in your lab you are already implemented the common average or a reference to your signal already, you can just click no here, so you don't need to load any data set. Just I will hit no, so I will show you how it will affect the data. The next thing that we'll ask is that if you want to change the slots, so reset slot boundaries, right now it just allocated one slot and with the 200 milliseconds. I will hit no here for now, and I will come back and look at it later. So if you just hit no, then it now started to loading the data set. So this is what you will get. If I zoom in here, so if you scroll on the y-axis, so I'm right now scrolling with my mouse to scroll. If you scroll on the y-axis, it will just zoom on that axis. If you, I'm sorry, x-axis. If you scroll on y-axis, it will zoom here. If you scroll inside of the plot, this will be zooming x and y. If you click on A here, it will just auto-range the thing. So I want to show you the effect of common average. So if I zoom in, you see this wiggly things all the way through. So this is because this signal has not been common average. So for this signal, the signal to noise ratio, this is a really good signal. It's still good, but let me show you if I reload the data set. So this is my data set. This is the median or common average for this data set. If I open this, now if I hit yes when I wanted to load the common average and give it this file, now I loaded the common average. Again, I will just bypass this boundary for now. I will get back to it later. So instead of hitting yes, I will hit no here. And I'm waiting. Okay, so now it loaded the data set and it analyzed it. So now if I zoom in, you can see all those wiggly things are gone, and it's a very flat and kind of almost noise-free signal. And so the signal to noise ratio is much, much, much better here now, okay? Now that we are here, sometimes when you zoom in, you can see that this dot is just sitting nowhere. The reason for this is that for the sake of responsiveness and speed, I'm subsampling this plot. So that's why it's not very smooth because it has been subsampled. But if you go to dissect module, then you should be able to see the raw data. Of course, the dissect module is a bit slower just because there are lots of data points in the plots that it has to plot. The main purpose of this plot is mostly just to get a bird's eye view of the whole thing. And If you want, you can zoom in, but most of the time you probably don't need to zoom in that much. But as I showed you, you can zoom in. So now going back to the beginning, we just loaded this data set. This is a data set that has LFP content and also high pass filter, both on the same channel. So these two filters, CS filter and SS filter, will be used to, CS filter will be used to generate the red trace. SS filter will be used to generate the black trace. And from the red trace and black trace, we detect the complex, from the red trace, we detect the complex box. From the black trace, we detect the simplest box, okay? We detected, we had this threshold here. You can also interactively change your threshold. And as you change it, you can see this line is also changing, or you can change it from here, or we'll change there. You can also directly go ahead and just change the numbers, and you can see this will change. For example, if I put this at, I don't know, 300, this will change and this will change. So this, and this are all interconnected. Similarly, the SS threshold here, SS threshold here, and SS threshold here are all, again, interconnected with each other. The point here, and if you click auto, based on some algorithm that I described in the preprint, it will detect a threshold automatically, and it will set the threshold automatically. The main point here is that always during the first slot, we just have one slot here, but whenever you load the data set, we are not going to have a template for aligning our complex spikes. Simple spikes are easy to align because we usually just align them based on either the minimum, based on this, or negative minimum, or the maximum, if I change this to the positive. But complex spike is a little bit harder. Right now, the way that this complex spikes has been aligned is based on aligned with respect to SS index. So if I have detected the simple spike before the complex spike, and then I just right after that simple spike, I detected a complex spike, and everything has been aligned to that. And because of that, you can see that not all of them are completely on top of each other. So the first thing that we are always do is that we are going to just define a template for the complex spike so we can align the data sets better. So for doing that, just need to get rid of the misaligned complex spike for now. So I'm going to hit select and then click, 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 click. So I just defined an area, hit OK, selected all of these. And if I hit delete, I will completely get rid of all of them for now, but I will redetect them. So if you look here, CS firing rate is 0.67. If I hit delete, it will go down to 0.3. It is as if I deleted them, but that was because I wanted to get the best spike. Now I hit learn template. So now I learned this template. You see this trace here. So this is the template that has been learned. Now that I have a correct template, and if you hit learn template, you can see that this will automatically change to align CS with respect to CS template. Previously it was on SS index, is now on CS template. And this is basically most of the time you want it to be like this if you want to work with the LFP content on the signature. Now what we are going to do is that we are going to reset everything again or redetect our spikes. And to do that, we just hit refresh. So each time that you hit refresh, everything will be reset to the beginning. So I will hit refresh, I will click that. So now that the software is redetecting all the simple spike and complex spikes, but this time the complex spike will be aligned with respect to the template, not with respect to the simple spike. And look, everything is now all in place, and basically it's what we want to see. And even we detected more complex spikes now, because now we had a better way of aligning them. So some of the complex spikes were lost because we didn't have any simple spike around it to align them correctly. But if you look here, for example, this, do you see this trace here? This is obvious that this is not a complex byte, and it should be deleted. And if you look at the PCA, this is presumably this one. So for selecting this trace here, you can either come here, select from PCA. For example, if I see, I should put you somewhere else. Select here, hit OK. So I select this. It will also show it up here, or you can directly select stuff up here as I did before. So I know that this is not a simplest byte, this is not a complex byte. It also shows it up here to you as well. So you can see what you have been selecting. Everything complex byte related will be in red. Everything simplest byte related would be in blue or shades of the blue. So I know that this is completely useless. So I just simply just delete it. Okay, that is gone, and that is gone from there as well. The other thing is that we had a couple of spikes here that I'm sure that these are just because we had a movement artifact at the end of this recording. So we want to also get rid of these ones because these are not signal, these are noise. So what we'll do is that presumably these are the clouds out here. So we can select them by the PCA plot. We can also select them, probably these are this shaded area here, or we can directly select them from the time point. So you cannot select from here, but you can, what you can do is if you put x-axis as time and y-axis as peak, this is basically these dots that have been plotted here are here now. So if I hit select here and then select those, then I just selected these points. Okay, and now I can hit delete. So that's what I will do. Okay, good. Now I got rid of those as well, and look at the suppression on simplest byte from complex byte. We kind of have a couple of things here. So let's say we want to see these things that cause this and decide if we should get rid of them or keep them. So what you can do here is that we had something here named time to previous CS. So this is time from the current simplest byte to the complex byte that happened right before the simplest byte. And if I zoom in here, so let me hit select so we can see bigger. If I zoom in here, you see these 2 points here? So there are a bunch that are at 0, and these are correct. But these 2 that are at around less than 10 millisecond, these are, if you look at them, these are not complex. These are not simplest byte. These are just basically kind of noise. And you can see them here that the peak velocity is less than the thing. So these are, just from the look of them, these are probably some spikelets of the complex byte that has been erroneously detected as simplest byte. So what you can do is that now we detected them. And these 2 are the ones that cause this bump here because I directly selected them to do that. So what you can do is just delete them. So if I delete them, what I expect to see is that this bump should go away. And if I delete those, this is indeed what will happen, that that bump is gone. And just for the sake of time, if you want to now go ahead and directly make sure that those 2 are in fact actually spikelets of a complex byte, let me just undo what I did. So you can hit undo here. So now they're back in business. And let me select them again. And so this 2 has been selected. Now we'll go to dissect module and to review them and make sure that I have to delete them or not. One way to look at them is just simply zooming in here and then just looking at them. So just zooming in here is obvious that this is a spikelet. So this is part of this complex spike. This is not a simple spike. But the faster way to review them is to go to the dissect module. And in dissect module, so right now the current one is SS. If I click next spike, you can see that this has been selected. And if you just click on zoom in, it will just go right on that thing. And as you can see, everything is more smooth here because this plot is not subsampled. So I'm looking at it. This doesn't look okay. So this is probably, this should be deleted. So since it is already been selected, you can just simply click delete spike and this will be gone, okay? As you know, we had, so let me zoom out here. We had 2 spikes here. So I can go to the next spike. So this is the second one and then zoom in and look at it. I just want to show you this, what does this auto zoom will do. If I click previous spike, this is not green anymore, but I stayed here. If I click zoom in, I will jump to where it was. So if you want to avoid changing and then click zoom in every time that you change your selection, if you just click auto zoom, it will always lock this window, this zoom window to where the selection is. So now if I click next spike, we'll kind of go with our selection. So we are auto zooming basically. If you don't want to do this, if you want to stay here, you can just uncheck this and it will not go with the selection. But each time you want to change the zoom window, you have to zoom in, out, or you have to directly change things here. Oh, one thing, still you can scroll here, you can zoom in and out, you can zoom in and out here. And if you, there are 2, if you can zoom in and out here with scrolling, there are 2 other things that always work with all plots. If you click left, click and drag and then move, this is panning. So you can just go over the signal. If you right click and move, this will be zooming. So if I right click now and then go that way, this is zooming in the x-axis, this will go up. This is zooming in y-axis. So for zooming, you can scroll here, scroll here, scroll here, or with the right click and then moving the right dragging, you can also change the zoom. So that's also one way to do it. Okay, so I have this spike selected. I want to get rid of it. I just click delete, gone. I go to previous one, zoom in. Sorry, this one is the one that I want. Select spike and then delete spike. Now it's gone. Okay. Once there is no selection anymore, so if I zoom out, if you click on next spike, it will just go over the spikes that are here in your recording. And so if I select an area here and hit select spike, basically I'm unselecting everything. Okay, so nothing has been selected right now. And if I click next spike, it will just start from the beginning, from the first spike. And if I go, we'll just go over all the spikes that are here. If I select something here, so let me clear ROI. If I select, by clicking, we'll just start the selection process. And if I select spikes, so now all of this has been selected. Now if I hit next spike, you see that these are getting things, and you can zoom in here. So now I'm just reviewing my selections. So that's the way it will work. So if you don't select anything, and if you hit next spike, it will go over everything. If you select something and hit next spike, it will go over your selections. Uh, right now, I'm reviewing simple spikes. If I click complex spike, now we'll change the mode. So now, since I have not selected any complex spike, if I hit next spike, it should start from here and should go through all of them. So there you go. Now this is selected. You can see it here. If I zoom in, auto zoom, now next spike, now I can just review all of my complex spikes. This is for the ones that are really careful about their data and wanted to look at each and every one of the spike to make sure that they have been selected here. Okay, and let me just mention one more thing. So let me select, so for selection, you can also select from this plot, or directly from here as well. So I just plotted something here. It selects spikes. So this two has been selected. Now let me select the first one. And so this is the green one. You can see it here. You can see it here as well. Let's say you are not happy with how this spike has been aligned. So right now we are using the alignment based on the template. So if I add a spike around something that has been already selected, I can effectively change the alignment for it. So I will uncheck the auto align because I want to manually align it. I will hit add spike, this will appear to show you where you're selecting. So I can align it, let's say here. So what I want you to pay attention is this green thing here, and that I have a spike here. And now if I do this, now this spike has been added. Basically, I just changed this green thing to a new spike here. So this is my new, and this changed. If I do it again, I want to align it here. This is just changing the alignments, okay? If you think that what was in the beginning was okay, just hit auto align, and we'll just use the template to put this dot wherever you want. Let's say you want to just completely get rid of this and make this a simple spike. If I change the mode to simple spike, and then now add something, so this is completely gone, that complex spike is gone, and now it becomes a simple spike. If you want, again, simple spike can be, for example, if you want to align your simple spike here, this is dummy, but you can put it here. Or if you auto align, no matter if you put a little bit off, since it's automatically aligning, it will use the tip of the thing. Or basically, if you want to do reverse, if you have a simple spike and you want to make it a complex spike, you can just put the current mode to the complex spike, hit add spike, and then hit here, and then it will automatically change that spike from a simple spike to a complex spike, okay? So that's everything with the dissect module. Probably I just covered everything. The other type of thing, if you just want to keep this, so let me select this. So this has been now selected. If you say move spike, it will actually just change this from a complex spike to a simple spike. So move the spikes, and it will just put it there. So you have to be careful because it will not realign it. So if you want to actually align it yourself, you had to do it, sorry. It's this, and then now it's a simple spike. Okay, any questions before I move on?
Just to make sure I understand, when you realign, it also removes the previous spike, right? It doesn't just add an additional spike. Okay.
Yeah, because so I have a couple of assumptions here in the preferences. You can see those assumptions. So the assumptions are that down here we had something about the conflicts. So we cannot have two simple spikes less than half a millisecond close to each other. So based on that assumption, if you click somewhere closer to half a millisecond with another simple spike, it will get rid of that and it will make a new simple spike for you. If your simple spikes are more than half a millisecond away from each other, then it will just add a new one. So it will not delete the previous one. We had another assumption here that we cannot have a simple spike half a millisecond before a complex spike, or half a millisecond again after a complex spike. So again, if your simple spikes are adjacent to a complex spike, we'll get rid of that complex spike. Or you cannot have two complex spikes less than 5 milliseconds close to each other. And this is just because the nature of the Purkinje cells, they cannot fire two complex spikes less than 5 milliseconds close to each other. But if you are 5 milliseconds away, it will just add a new one. So do you see? So the definition of what is around something or what is adjacent to something is based on these numbers here. And if it is not around, it will just add a new one. If it is around, it will just replace that one and put it in a new position. And you can also change these numbers. This is just default values. So you have access to basically everything in this software. You should be able to access that. Okay, so this was a kind of very good session that I have a very high quality data. I have my LFP content and I have my the actual signal. Let me now move on to a session that does not have the LFP content. And this happens a lot, especially with the Cambridge recordings. This might happen. So this is a session that now those peaks that we saw in the LFP content, they're no longer exist. But I know that this session has simple spike and complex spike. So this is where the magic of the UMAP will come into play. So what will happen is that I can bring this down kind of and start. Go ahead.
Sorry, can I just ask a question regarding the previous one? So let's imagine you have multiple slots and you are doing a learning paradigm, right? And basically the excitability of the Purkinje cell is changing over time. And so you might imagine that you will have a number of spikelets that might change over time too.
Yeah.
Would you be able to catch them? Or you would need to basically cut in very small slots to make sure that it change smoothly over time, or what would you recommend if it was the case?
So we have those scenarios. Actually, let me show you the next data set. It will have something like that. So
okay, perfect.
See. So let me go to this data set. The short answer to your question is that usually for those scenarios, the first spikelet is time locked. The spikelet 2, 3, and 4 are the ones that are jittering in time.
Yeah.
And when you define your template, it usually has time lock to the first spikelet, and it has an average of the second, third, and fourth spikelet if you have. And that first spikelet is enough to give you a good alignment. And this was the case in our recording. We also included a sample data set that has multiple spikelets in the paper. In figure 2, we included that. And we were able to perfectly, just using the template was enough to detect those things. But as I mentioned, if you are not happy with the alignment, you can definitely select those and go back and manually change the alignment. Right now, we just use one template. If you want to be super fancy, maybe you might want to improve the software because this is open source, and maybe define 2 templates and then align with 2 or more than 1 template. You can also do that, although currently we don't have it. But if you're interested, you can actually improve the software and do those type of things as well.
Not yet. It was just a question for now.
Yeah.
Thanks.
Okay. So here I have a recording that if you look at the PCA, right now, if you just look here, you can see that I have the main signal here, a bunch of, and these are noise probably down here. So when I'm looking here, I have the main signal here, I have a cloud out here, and I have another cloud out here, okay? So because I already know the answer to my question, this is noise, and this is the complex spike, and this is the main signal. But in the PCA space, there is no clear cut between the main signal and the complex spikes. But if you go, instead of PCA, if you go to UMAP, then that's where the magic happens. And then we will be able to select these things much better. So let me just select them in PCA space. So if I select them in PCA space, this is basically what is going on, but you can see that it's not very clear. So let me unselect everything. UMAP by default is deactive because it's computationally expensive and it will slow down the software. So if you want to activate the UMAP, you have to go to Tools, click on UMAP for dimensionality reduction, and then you will activate the UMAP. So this tick is now here because the UMAP is activated, and just automatically will use UMAP instead of PCA, but still I have access to PCA, and now I can also select UMAP as well. So let me just select this cloud here. So these are my complex spikes, okay? And why I'm saying these are my complex spikes, because if I move them to the complex spike pool, that I will do, you will see that it will cause suppression on the simple spike. But I want to first show you how UMAP will help with the PCA. So these are my cloud of complex spikes. You see these couple of spikes here or a couple of spikes here, that if I just work in the PCA space, I will not be able to detect these because these are mixed with the actual signal, with the simple spikes. But in the UMAP space, this is the kind of power of the UMAP, that they are all out here. So I can directly select them and it's very clear. So now I have selected those. I will say move to CS. So now it will just move these. To the CS here. One thing that Guy might be kind of answering your question is that these complex spikes that we have here, so let me go here and then looking at them, some of them has this extra bump, some of them does not have that extra bump. You see that? And if I now I don't need to form the template because I detected my complex spike from here to here. So they are already being aligned based on their simple spike content. So you don't need to, when you bring a complex spike from here to here, you don't need to form the template, and you don't need to refresh. But I'm just doing this to show you what will happen is that when I'm forming my template, because I have a bunch of complex spikes that have the second bump up here, some of them does not have the second bump. Basically, the template, as you may expect, is the kind of the average of those two. So it's in between. But since the first bump are always the dominant one, this is enough for the alignment. If the case was that we had something here. If you wait for it, I have another example that will kind of showcase that the template is enough in most cases for those things. And what you can see here is that now I have this, and then I have a big bump here, and then another thing. So just based on experience, I know that this is the noise. These are these things and that have been erroneously detected. But just looking here, it's obvious that this is probably not belonging to the signal. So we had to take care of this. So we had to hit select. We go select these ones. And These are, as you can see, they're all up here. And if you look at the waveforms, they are not look okay. So if I delete those, because they have been deleted, this also took care of that big bump here. So now I have a healthy simple spike autocorrelation and a healthy cross correlation between complex spike and simple spike. So just to, again, review, for these data sets, because I didn't have that LFP content, I just brought down my threshold to a low value. I detected everything in the simple spike pool. Then using the UMAP, I detected my complex spike and then moved them to the complex spike pool. And then I confirmed that these are complex spikes by looking at the suppression from the simple spike. So based on these two, I know that these are simple spike and these are complex spikes. Is that clear? So I will just show you one more example. And this example is a good one because this is the Purkinje sort challenge data set. This is a data set. And so this data set already has the common average. So it's already being common average. So I don't need to load the common average. So I'll hit no here. I want to just showcase the slot boundaries for you. So I will click yes here so we can take a look at the slot boundaries. So this is a really interesting data set because it has two simple spike and two complex spike at the same time on one contact. So because I hit yes on the slot boundaries, we come here. So here you can change the number of slots. So right now I have 3 slots. You can click them and move them around. For example, I want this slot to be a little bit just right after this probably movement artifact. You can also manually add a new slot if you like. So you can just directly hit add line at click and then add a new slot. If you change the number, what it will do is that now it will uniformly distribute the slots for you. Okay, and then hit OK, and then it will now start recording with 5 slots. So with the examples that we had so far, we mainly had just 1 slot, but right now we have 5 slots. So what you will do is that once you are done with the first slot, you have to go to the click next. So you have to go to the next slot, analyze that, go to the next slide, analyze that, and so on and so forth. Again, since we had LFP, we had to form a template and then hit refresh again. So the complex spike will be aligned based on that template. So what I will do is that I will hit select, get rid of these misaligned complex spikes. Of course, I will redetect them. You see we had 2 types of complex spike here. This is one type, the other one is this one. And when I hit template, it's kind of is average of them, kind of. So hit refresh. Now I have all of them. So it's 2.1. Presumably 2.1 is too high for a complex spike firing rate. Also, if you look here, you can also see that I have kind of 2 clusters here and 2 clusters here. So let me activate the UMAP and then actually close this down so we'll have a better view of. So I clearly have 2 clusters for my complex spikes. Oh, one more thing, you can always change this thing and change the window of time that you are including in your analysis. So sometimes you want to just, let's say, focus here or focus out here. I should have done that with the previous example. Anyway, okay. So now the question is, so I can see I have 1, 2, 3 kind of clusters here and 2 clusters here. Which of these simple spike and complex spike are corresponding with each other? So we had this new module and kind of cluster module that is all courtesy of Amin. that he made this, and he's working on this to expand it for the multi-channel reporting stuff. But with the cluster module, now we can very easily see which one is associated with which one. So if I click on cluster, now I'm in the cluster module. My current mode is complex spike. If you change this to simple spike, you can look at the simple spikes. If you change it to complex spike, you can look at complex spike. So I want to, right now everything is labeled zeros, so everything is just one label. For assigning a new label, you can directly select stuff. It selects spikes and set label. So this was label 0. Now we'll make it label 1. So now this is black, this is blue. So I have two labels now, and you can see the distribution down here. You can see the waveform for label 0 here, or waveform for label 1 here. Okay, see that they're a little bit different from each other, actually. Okay, but now we also had to label the simple spikes as well. So you can again directly select them, or you can use the automatic out methods. We had Gaussian mixture model, we had ISO split, we had HDBS scan. Let's use Gaussian mixture model. It apply how many clusters do we want? 1, 2, 3, 4, 5. So I need 5 clusters. It will ask us to select the estimate for the center. So 1, 2, 3, 4, and 5. And there you go. So interestingly, when I select this, it kind of expanded to the 3 here. So maybe instead of this, because I know that I want this, I will hit, I have to do it the other way around. Sorry, I have to select these ones, select spike, set, and then assign them to label 1. So now all of these are label 1, okay? So now what you can do is that, again, you can see everything, but I can start crossing these labels with each other. So if I have label 0 for complex spike, and let's say label 2 for simple spike, this is definitely a simple spike, but it doesn't have a suppression. What about label 1? Look, label 1 and CS cluster 0 have a suppression. So these two are corresponding with each other. What about the other label of complex spike? So this one definitely does not suppress that, but what about label 2? So when we look at label 2 is suppressing the other complex spikes. So this is confirming that one cluster of simple spike is suppressing, one cluster of complex spike is suppressing this cluster of simple spike, and another cluster is suppressing this one. What about the other ones? So let's say, what about label 0 here? So if you look at label 0, it's basically probably because we can see the distortion here. This is the noise. What about label 3 and 4? So label 4 is getting suppressed from complex spike 1 and also 0. So this is the overlap between simple spike 1 and simple spike 2. So this label 4 is both simple spike 1 and simple spike 2, because these two signals can also overlap with each other. But the point is that right now, the way to use this P-sort is not supporting multi-contact or multi-cell sorting at the same time. So although I have assigned all these labels, and now I have a very good idea about what is what with my simple spike and complex spike, if I hit OK, what will happen is that, so I hit OK right now. What will happen is that all those labels are gone, and the main window, this is where things are matter. I just have one label, and that's complex spike, and there's 2 clusters, all of them are complex spike, and everything here is again simple spike. So the way to unfortunately work with the software for now. We're working on to solve this issue is that you had to keep one Purkinje cell at the same time. So you had to keep, let's say, Purkinje cell 1. So I have to delete everything else and then sort the data till the end, reload the data, and then sort the Purkinje cell 2 until the end. Usually this is a very kind of special case that I have two Purkinje cells on the same contact. So usually the scenario is that you have one cell per contact and you don't need to resort your data each time for each cell because you just have one to begin with. But the way to do it, and let me just quickly go back. Let's say I like to keep label 0 around, so I will this label 1 select and then delete spikes. So just get rid of that. Let's go to simple spikes. And then let me come here, select spikes. I know that these are noise, so get rid of them. Then label 2 is the one that is not suppressing my complex spikes. Select spikes, delete spikes. And this is where we're left with. So there we go. So now we had our simple, so all of these are our simple spikes. And this is a simple spike. And all of these are our complex spike, and this is our complex spike. And this was all just slot 1. And we have 5 slots. So now we had to go to the next slot. And then again, this was kind of start over. But going to the next slot, it already will bring the template from the previous slot. So you don't need to redefine your template. It already has it. If you want to change it, you can unlearn it and relearn it. But if you're happy with your template, you can just use this. This is also really helpful because when we have drifting in the signal, sometimes this template will change. So the whole purpose of this slotting mechanism is to account for the drift in the signal. So as the signal is changing, we can just adjust our parameters and sort each slot, fine tune our parameters for that slot. So we'll have the best results possible. I will stop here.